All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me here today is my colleague at NCCAT, Ed Eng. Joining from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt. And from the Pacific Northwest Cryo-EM Center, PNCC, is Theo Humphreys. This webinar is a regular event that we host the last Thursday of every month, highlighting methods, tips, and tricks that groups are using to obtain and interpret the data they can collect using National Center resources. So you can already save the date now for our next talk on October 27th, when Robert Monson from the University of Louisville Medical Center will give a talk titled From Meme to Model, How Cryo-EM Helped Resolve a Misconception in the DNA G quadruplex field. So if today's talk on RNAs turns you against them, come back next month and we'll talk about the deoxy counterparts. And actually, coming up in December, in place of our seminar format, we're going to host a roundtable discussion with a panel of users with lots of time for more interactive Q&A. We're still deciding the exact topic, but look out for announcements about our December roundtable. So today, Theo will introduce our speakers, Stephen Bonilla, after my colleagues from each of the centers give a quick update about the resources we make available at no cost to the research community. And we are recording today's talk. You can find the recording along with recordings from all of our past talks on our YouTube page or at the events page at cryoemcenters.org. And one last final bit of Zoom logistics, we're going to watch the Q&A box for questions. So please use that and not the chat. And I will be moderating questions at the end of the talk. Unless there's something very specific, we may interrupt Steve with a question or two if it's very relevant during the talk. So with that, I'll turn over to our center representatives and I will ask Mike to go first.
Good morning, I'm Mike Schmidt. I'm one of the co-directors of the Stanford SLAC Cryo-EM Center, located at SLAC and up near the Stanford campus. We have 3 and soon 4 Krios microscopes and all of the specimen preparation material needed, Vitrobots, Vitrojets, chameleons. We will have them all by the end of the month or by the end of the year. So please come. You can send your already clipped grids, or you can come in person and use our specimen preparation for screening, for training, and for high resolution data collection. Please come see us.
Thank you, Mike. Next, I'll turn over to Ed.
Hello, my name is Ed. Here in New York City, where NCCAT is located, we have high-end data collection on 4 dedicated Krios instruments. We have a robust grid preparation and screening program with blot-free vitrification using chameleon and a glacios microscope and a cross-training program. I would like to point out, if you're coming from an idea state or underfunded region, you can talk to the NCCAT user office about coming on site, learning about proposal writing, as well as subsidized housing. I want to point out that in the spring, we're bringing back our short course series with a tomography short course with our sister center in situ. So learn more at nccat.nysbc.org.
Thank you, Ed. And I'll turn over to Theo.
Hey, my name is Theo Humphreys, and I work as a microscopist and scientific point of contact at Pacific Northwest Cryo-EM Center. Here we offer 2 proposal types. These include a screening access mechanism, which allows for 5 days of on-site sample preparation, optimization, which is targeted at low resource institutions. We also offer a general access proposal for single particle or tomography projects. These offer up to 480 hours of microscope time a year for up to 2 years, and also allows you to be eligible for many of our workshops. Each approved proposal will also be assigned a scientific point of contact, such as myself, who can help you through your scientific and Cryo-EM workflow. At the PNCC, we have 5 microscopes, including an Arctica, and 4 Krioses, all of which are equipped with K3 detectors. We also have a Krios with a Falcon 4 and 3 Krioses with energy filters. We also offer a VitraBot and Leica GP2 for sample preparation, and we hope to hear from you soon. And you can find out more at our website. And with that, I'd like to introduce today's webinar speaker, Steve Bonilla, whom I've had the pleasure of working with at the NCC. Steve is currently a postdoc in the Keefe Lab at the Colorado Anschutz Medical Center. And today I'll be discussing his lab's work using Cryo-EM on structurally and conformationally dynamic RNAs.
Great. All right. So Theo, can you hear me and can you see my slides? So good?
Yep, looks good.
All right. Well, yeah. Well, thanks for the introduction. I'm Steve Bonilla. I'm a postdoc in the Keefe Lab. And today I'll tell you how we are using Cryo-EM to visualize conformationally dynamic RNAs that have been difficult to characterize by other methods. All right. So I'm sure that you all know that RNA molecules form intricate three-dimensional structures to assemble with proteins to form this large RNA-protein complex machines, such as the ribosome. But what some of you may be less familiar with is the fact that there is a vast number of other functional RNA three-dimensional structures that catalyze reactions, bind specific ligands, and interact with specific proteins to play very diverse functions in biology. And like proteins, these RNA molecules depend on the formation of specific three-dimensional shapes to function. However, in contrast to proteins, for which we have tons of experimentally determined 3D structures that teach us about protein structure function relationships, the field of RNA structural biology is still in its infancy. And for a rough comparison, in the PDB, the number of RNA-only structures is about 1% that of proteins. And there are several reasons for this, but one of the major reasons is that RNAs present certain experimental challenges that I'd like to discuss next. So the basic unit of RNA 3D structure is the A-form helix, which is stabilized by highly local interactions, hydrogen bond and base stacking interactions between neighboring bases. And to form or to build a functional structure, many of these modular helices come together in 3D space to form larger assemblies in which these helices are connected to each other by a relatively flexible junctions. The overall tertiary structure then is stabilized by the formation of a sparsely distributed tertiary contacts. So in contrast to folded proteins that are stabilized by a large number of tertiary contacts and by the hydrophobic effect, RNA tertiary structures tend to be less compact and stabilized by fewer tertiary contacts, resulting in highly dynamic tertiary structures. So it is more accurate to think of these structures as dynamic conformational ensembles that fluctuate between multiple conformational states. And this dynamics, these fluctuations are often important for the function of the RNA, but they have the consequence of making RNA structures very challenging targets for structural biology. Now, as many of you know, recently, Cryo-EM has emerged as a very powerful tool that in some cases is able to solve the structure of relatively dynamic macromolecules. And one of the main advantages of Cryo-EM is that having the capability of imaging single particles, in principle, it is capable of capturing images representing the same macromolecule in different conformational states. And that, of course, allows you to separate the molecules based on their conformation and allowing you to refine them separately, potentially to high resolution. Now, in addition, the conformational variability captured by Cryo-EM can give us some qualitative information about conformational dynamics. So today, I'll give you a couple of examples in which we were able to use these tools to solve the structures of conformationally dynamic RNAs. And I'll discuss a bit the process that I follow so that maybe that can be useful to some of you. And I'll also mention a couple of computational tools that make them handy when dealing with RNA molecules. All right, so in the KEEP lab, we are interested in understanding structure-function relationships in RNA viruses. And from an RNA structure perspective, RNA viruses are fascinating because they have evolved to maximize the information content of their relatively small genomes. And they do this in part by encoding multifunctional three-dimensional structures that allow them to hijack the biochemical machinery of their host. And one way in which they can do this is by mimicking the structure of cellular molecules. Right? So given the importance of tRNA, which I think probably all of us are familiar with, it is not surprising that RNA viruses have evolved ways to mimic tRNA structure. And there are several examples of this found in different viruses, including in some that infect humans. But the best known examples are the so-called tRNA-like structures, or TLSs, that are found at the 3' end of certain viruses that infect plants. So these structures were originally discovered by their ability to recruit a specific host aminoacylated synthetases to add specific amino acids to the 3' end of these structures. And they are also recognized by other tRNA-specific machinery of the host and are important for many aspects of the viral life cycle. And a very interesting example from a structural perspective was this variant from the brome mosaic virus. So this is a tyrosine-specific TLS. And I'll focus on structure, but first I would like to discuss just a couple of things about its function to put things into context. So like I said, this structure is found in the brome mosaic virus genome. So the brome mosaic virus, BMV, has a tripartite genome. So its genome is encoded into 3 separate RNA molecules. And each of these molecules contains one of these TLS structures at the 3' end that gets charged with a tyrosine by the tyrosyl-tRNA synthetase from the host. This structure is conserved across viruses that are related to BMV. And it's been shown that this structure and in its aminoacylation results in enhancement of the translation of the viral proteins. However, the mechanism of that enhancement is not very well understood. And like I said earlier, this structure also appears to interact with other tRNA-specific machinery of the host. In addition to those tRNA-like properties, this TLS structure contains the sequences that are recognized by the RNA-dependent RNA polymerase during synthesis of the minor strand during replication. And also the TLS structure serves as a nucleation site for formation of the capsid that encapsulates the genome. So obviously this structure is very important for the virus, but many of these processes, the structural basis of many of these processes are not very well understood because we didn't have a structure of the TLS. And particularly puzzling was its ability to mimic tRNA. And that was very interesting because it was not understood how its much more complex and larger secondary structure could be organized in 3D space to mimic the much smaller, simpler tRNA. For example, we didn't know whether the TLS was able to form something that resembled the canonical L shape of tRNA in 3D. We knew that the TLS is able to interact with the tyrosyl synthetase homodimer to get tyrosylated, but we didn't know whether those interactions resembled those of tRNA. It was still a question whether there was something that resembled or acted as the anticodon arm of tRNA in the TLS. In addition, we had very compelling evidence that this TLS is conformationally dynamic, such that it evaded crystallization for many years. But we didn't know whether those dynamics were related to its function or its ability to mimic tRNA. So we decided to pursue these questions by Cryo-EM, even though the RNA is only 55 kilodaltons, so it's a very small molecule relative to the conventional targets of Cryo-EM. And it's also dynamic, so that also presented a challenge. But after some optimization of conditions and particle concentration and such, we're able to get some nice grids that we took to PNCC. And this is me and Theo here right before the pandemic. We didn't know what was coming. This is just like a month before the beginning of the big of 2020, and this is at PNCC, and Theo and the PNCC team helped us get this very nice Cryo-EM data of the TLS. And something that I'd like to point out at this point is how high the contrast is of these RNA molecules, even though they are small. And this is related to the very high electrostatic potential of the RNA backbone, which produces this very high contrast. So at least from a particle picking perspective, RNA seems to be a bit easier relative to proteins of similar size. All right, so at this point, I would like to discuss a little bit of the process that we follow to get a map of this RNA. So something that we found very useful in the process was to follow this. Okay, I'll show you this little protocol that we follow, in which so after a very limited 2D classification, so at this point we still have a lot of the particles in our sample, we generated ab initio reconstructions, and consistently only one of these ab initio reconstructions display features that were that resemble RNA secondary structure, which is helical features. So then we performed a heterogeneous refinement, which is a type of 3D classification to classify the particles that match this. Let's call this the good volume, right? The one that display the secondary RNA secondary features. And then we went through this iterative process with where the particles that classify with this were again reclassified, that were put together, were put again into this process, and the particles that classify with this, let's call them bad volumes, were discarded, and we kept just keeping the particles that classify with the good volume. And we did that a few times, so that has the effect of essentially selecting the subset of particles that best match your reference volume. And this is, of course, something we didn't come up with. This is something that we got from the CryoSpark forum, which is a very useful resource that if you are not using, I highly recommend that you go to the CryoSpark forums. There are many questions there that are answered all the time. All right, so then we took that set of particles, we performed homogeneous and non-uniform refinements in CryoSpark, producing this very nice Cryo-EM map of the TLS. And let me remind you that this RNA is only 55 kilodaltons, and it had been elusive to structural biology for a very long time. So at this point, we were already very happy with this result. This is the first time that we see what this RNA looks like. But to fit the structure into this map at this moderate resolution, we decided to get a bit more information. And the way that we did that was by using this strategy in which we took this is the known secondary structure of the RNA, and we designed this variant, this mutant, in which we extended or truncated specific helices of that secondary structure. And then we took Cryo-EM data and looked for changes in the density that were consistent with those modifications. And that allowed us to map essentially the secondary structure into the 3D density. And this is something that can help you either give you additional information when you are modeling the structure or testing your model, your structural model. And this is allowed by this very modular architecture of RNA that allows you to make this modification without altering the overall fold of the RNA. All right, so we use that information on the original map to get a structure of the RNA, and just by comparing the TLS to genuine tRNA and knowing how tRNA interacts with the tyrosyl synthetase, we were able to hypothesize that this arm B3, I'm calling this helix here, is acting as the anticodon arm. And we then, using the structure, we were able to verify that by making targeted mutations and aminoacylation assays. And this aminoacylation assays supported the hypothesis that this B3 acts as the anticodon arm. However, when we modeled how this RNA would interact with the synthetase, we found that in its current conformation, the RNA wouldn't fit the binding site of tRNA. There will be this big clash with the protein. So that suggested to us that perhaps the RNA was not pre-organized for this interaction, and that it may need to undergo conformational changes. And consistent with that model, we did this 3D variability analysis in Cryo-Spark, which gives you some sense of flexible regions or relative dynamics. And that 3D variability analysis was consistent with this anticodon arm analog being relatively flexible, and opening the possibility that it could potentially rearrange to accommodate those interactions and avoid the steric clash. But to test that model more directly, we went ahead and reconstituted the complex of the TLS bound to the tyrosyl synthetase. And it was very nice to see, even in the 2D projections of this RNA, of these complexes, how the complex of the TLS with the tyrosyl synthetase resembled the legitimate tRNA complex. So here on the right, I'm showing you this is the crystal structure of the tRNA bound to the synthetase. The synthetase is a homodimer, and it can bind 2 RNA molecules. And you can see how that resembles this 2D class here of the viral RNA or the viral tRNA mimic, each of them making 2 interactions with the protein. At this point, we ran into some problems with preferred orientations that we were having with these complexes. But then we were able, again, at PNCC, we took data at different angles, so tilted data, and that allowed us to eventually get a 3D reconstruction of what the RNA looks like when it's bound to the synthetase. And consistent with our hypothesis here, this anticodon arm analog has to undergo this very large rearrangement to avoid those steric clashes and bind to the protein. And something that was surprising was that the TLS, the viral RNA, did not need to assume this L shape of tRNA, and instead binds with a very different geometry, in which this acceptor arm and the anticodon arm approach the protein with a near parallel orientation. So that was very interesting, and it opens the question of how many other ways are there to interact with tRNA specific machinery, and there may be many other viruses or other RNAs that may be using this this machinery in unpredictable ways. All right, so another finding, another interesting thing that we found was when we were looking at our complexes, where that we found more than one bound state of the the RNA, and these different states differed in the degree of interactions between the protein and the RNA, and this suggests a multi-step binding process, right? And also suggests that this complex itself may be conformationally dynamic. So in contrast to tRNA that appears to be largely pre-organized for this aminoacylation, the viral mimic exists as a dynamic conformational ensemble in which this anticodon stem undergoes it probes many different, it samples many different conformations, and then it undergoes this very large rearrangement that is stabilized by interactions with the synthetase. Our data also revealed multiple bound states that suggest that this is a multi-step binding process, and that this complex itself is conformationally dynamic. So this is the end of this first story that I have about this viral RNA. And the rest of in the next half of my talk, I'm gonna switch to a different story. So I guess I should ask if there are any questions, or maybe I can just continue and wait for the end. But if there aren't, I'll just switch to.
I don't see any questions that have popped into the box, but we'll give people a second. Here comes one.
Okay.
Carlos Kikuti asks, are you still trying to improve resolution of TLS alone? If you are, what are the most promising ways? And if you're not, at which point do you give up? How do you decide when you're done trying?
Yeah, that's a very good question. Yeah, so yeah, we at this point we are not we left the TLS at that point. Instead, what we've been trying to do is to look at other variants. So these TLSs are spread across different viruses, and they have different architecture, secondary structural architecture. So it will be very interesting to see what those RNAs, how those RNAs basically accomplish the same job using a different secondary structure architecture. And it's possible that some of those RNAs may resolve to high resolution. Maybe they're more rigid. How will I go about increasing the TLS resolution? I don't know. I mean, I'm not completely sure if getting more particles potentially could, and doing like larger or more extensive classification could result in higher resolution. But yeah, I think that maybe getting more data and trying newer versions of classification could potentially lead to high resolution.
Yeah, a couple more questions have filtered in. So Chiara Rapsidara asked, did you try any other ways to improve the orientation distribution of the particles, or did you just do tilted collection?
Yeah, I think that we just, I went straight into tilted collection. And yeah, I think, I mean, from talking to people, I think probably the best, I think that the best way to do to approach this will be to try to fix those preferred orientations in some biochemical way, or some changing the solution conditions before you go into tilting the data, tilting the grid. But that's how we did it. We just went straight and tilted.
Yeah, and there was a question about how you chose the tilt to use for that data collection.
Yeah, so I think so. I don't recall the name of a little program that we used. It just escaped me right now, but I'm sure that you can look it up. There is a little script that you can run on your, I believe, on your reconstruction that tries to find what will be the optimal angle which you and even tells you, I think even tells you the proportions of data that you should take. And I think that we
is it Cryo-EF?
Yeah, that sounds
okay,
that sounds
a
right.
couple people chimed in, so
yeah
thanks to
I
the
think
audience.
so. Yeah, thank you, audience. Yeah, so yeah, I think that we use that, and that recommended I think that recommended that 20 degrees. But then after discussing with the PNCC team, we decided to also collect at 40 degrees, I believe. So so we did 2 different angles. Yeah.
Samuel is asking, even though you have good contrast,
Yeah.
did you still use a phase plate or energy filter for the data collection?
The data has an energy filter, but we did not use a faceplate. So no faceplate.
All right. Minwoo Sung asked, for the 3D variability analysis, were you able to find which motions are most significant from probability principal component analysis? I assume there's some random motions captured from the variability analysis as well.
Yeah. So so we so that variability analysis allows you to choose how many components you solve, basically, right? So it gives you and those components are presumably capturing where the most variability is, as in principal component analysis, and consistently across the different components, we always saw that the area that I show you, that was the most variable, right? So that was always moving. But yeah, there were other areas of the RNA that were also, according to the analysis, also moving a bit. But those motions were smaller, and we did not focus on that. Yeah.
All right, and there's one more very specific to this part of your talk, and then we'll let you get back to it. But does the tRNA synthetase change its structure even a little bit in order to interact with the TLS? Did you observe any difference in the interaction with TLS?
Yeah, those are very good questions, and that's something that unfortunately at the resolution that we are, because these are still relatively low resolution, it wasn't sufficient to see any conformational changes with that I would say we will be confident to tell. At this point, at least the protein was basically just, we docked I think we docked, and there was some refinement that was done, but this is something that I'm really interested in doing, is to increase the resolution of the complex and see like really nail down what those interactions are between the TLS and the enzyme, and also whether the enzyme is undergoing rearrangements to accommodate those interactions.
Awesome. Yeah, and actually, I had a question. What do you think is limiting your resolution for that structure? Because it seems much lower than the TLS on its own.
Yeah, I think that there's probably a lot more room for the preferred orientations were definitely one of the there I showed this so the preferred orientations. There's also the this variability that we saw, right? So I showed that there were like multiple bound states, and there is the there's also the combinatorial thing there, because the enzyme combined two different RNA molecules, and we believe that those two RNA molecules can be in different states, right? So then you can have multiple combinations of them. So they're maybe collecting more data and doing much more stringent classification could potentially lead to higher resolution. For the purpose of this story that we were trying to address, we basically we're focusing on this conformational changes of the RNA, and at this resolution that was very, very clear. But yeah, I think that to get more details and even like maybe some mechanistic insights, getting high resolution of this complex could be the way to go.
Actually, there's one more question.
Mhm.
So I'll throw it to you before you move on. What concentration of RNA sample is good for cryo-EM?
Yeah, so we found that that the concentrations tend to be higher than what you will use for proteins. And I will usually start with like 10 micromolar. So you have to do the conversion there to your, and these are like small RNAs, so this is 55 kilodalton RNAs. 10 to like 50 micromolar of this is what we used, which I believe is relative to proteins of similar size. I think it's a higher concentration. Yeah.
All right, we made it through the questions for that part, so we'll let you move on. I'm sure more will filter in.
All right, sounds great. Thank you. Okay, so all right, so let me now switch to the second story that we recently published, and that story is about RNA folding and the RNA folding problem. So before we were dealing with folded RNA structures, now we're gonna look at the RNA folding problem, and that is to try to understand how a transcribed RNA sequence, right, finds its native folded state out of a vast number of possible conformations. And in practice, the way that the field has approached this problem is to find a RNA folding model system, and then detect or use a wide array of techniques to detect and characterize different conformational states that precede the formation of your native folded state. And many techniques such as small angle X-ray scattering has been very useful, time-dependent chemical probing, thermodynamic and kinetic analysis. All of those tools have been very useful to elucidate important aspects of this folding process. And what was very clear, even at the beginning, was that RNA folding landscapes can be very complex. And RNAs have a tendency to go on these alternative folding pathways that may result in the formation of alternative stable states that can have new functions, or can be misfolded states that trap the RNA in a non-functional state. Right? So ideally, to understand how an RNA folds or misfolds, we would like to have 3D structures of these intermediate states, ideally to high resolution. But as you can probably tell by my question marks here, in most cases, we don't know what these intermediates look like. But a lot has been learned indirectly about folding and misfolding, even without solving these 3D structures. And so something that we learned is that RNA folding is a very hierarchical process that typically starts with the formation of secondary structures, and that basically the formation of helices, which I'm representing here with these cylinders. And there can be multiple intermediates that differ in the degree of secondary structure formed. And then those helices come together in 3D space to form tertiary contacts that stabilizes tertiary structures. And going back to the formation of the secondary structures, we know that these secondary structures are stabilized by the formation of base stacking and base pairing hydrogen bonds. Now, something that we need to keep in mind is that although canonical Watson-Crick base pairs are, what we usually think of when we think of base pairing, and they are very important, in reality, there are a large number of other possible combinations of base pairs. And essentially, any base can make interactions with any other base. So you can see from here that a consequence of this is that the native secondary structure has to compete with the formation of many other possible secondary structures, some of which can be as stable or very similarly stable as the native secondary structure. So from here, then you can see how during the folding process, there is this thermodynamic tendency for the formation of alternative secondary structures that may lead to trapping the RNA into this alternative non-functional states or alternative functional states that I was referring to earlier. A lot of what we know about folding of RNA has been learned from the study of a specific model system of an RNA functional structure, and that is the group 1 introns. So I'm just going to give you a little, I'm going to just tell you a little bit how this structures work. So this group 1 introns, they fold into this 3-dimensional structures, which I am representing here by this squiggly line. And these 3D structures contain a binding site for an exogenous guanosine residue that then serves as a nucleophile in the first step of a couple of reactions, of chemical reactions that essentially at the end result in the excision of that 3D structure and the joining of the flanking exons. And of course, this is a very simplified schematic of a much more complex process. So like I said, to do this, the group 1 intron folds into a complex 3D structure that in our cartoon cylinder version looks more like this. This is a cartoon version of the tetrahymena thermophila ribozyme. And here in blue, I'm gonna keep putting this cylinder in blue here, or this area, which is where the guanosine binds. So this is where the active site is. And recently, very nicely, the groups of Rijudas and Wachu solved the structure of the full tetrahymena ribozyme by Cryo-EM to 3.1 angstroms resolution. The core of the ribozyme had been solved previously by crystallography, but the peripheral domains surrounding the core, or some of them, were very dynamic, and therefore they were not they couldn't crystallize them until these groups came and used Cryo-EM for this. All right, so we're talking about the folding of that structure. So how do you get to that structure in the first place, or how can it misfold? But before I do that, I want to give credit to some of the people that through many years of work figured many of these things out, even without having 3D structures. And the project that I'm about to show you builds from their work. So many of the models that I will discuss are based on biochemical and functional studies done by Rick Russell when he was in Dan Hirschlag's lab, when he worked as a postdoc in Dan Hirschlag's lab, and later work done by David Mitchell III when he was in Rick Russell's lab. And a lot of this work was based on previous observations made in Sarah Woodson's lab. And of course, there's many other colleagues and other groups that have been building our understanding of RNA folding through the years. All right, so going back to the folding of this tetrahymena ribozyme. So a series of early experiments determined its folding pathway and found that under standard experimental conditions, most of the ribozymes go into this misfolded state that has no catalytic activity. Right? So just like those misfolded states that I was referring to earlier on my RNA folding schematic cartoons. And then they also found that then the transition from this misfolded state into the functional state is very slow. So from these observations, the simplest model was that this misfolded state is just some very different structure where the RNA is trapped, and that comes from the observations that first the misfolded state is inactive, and then it requires a lot of refolding or structural reorganization to go from the misfolded to the native state. So that suggests that this misfolded state is very different from the native state. Now, Rick Russell and colleagues then made some very interesting observations that presented a paradox. When they use different biochemical or low resolution structural tools to compare the native to the misfolded state, they found that they were essentially indistinguishable. So they use chemical footprint or chemical probing to get these footprints that were very similar. And that suggested that these two states, they essentially form the same secondary structure, and they form the same long-range tertiary contacts. And they did some mutations that also were consistent with the misfolded state forming the same set of tertiary contacts and the native state formed. And then they did SACs, and they showed that the SACs profiles were very similar, although there were some differences that suggested that the misfolded state is a bit less compact than the native state. So that presented this puzzle, because then, given these similarities between these two different states, what is the structural basis for the catalytic inactivity of the misfolded state, and why will it take such a long time for one to refold into the other? So that is this paradox that I referred to earlier. And again, Rick Russell, Dan Hirschlag, and colleagues came up with this very clever model that essentially is consistent with all these observations. So in this model, the native and the misfolded state essentially form the same structures, but somewhere near the active site, there is a pair of incorrectly crossed single strands that are undetectable by biochemical methods and cause these functional differences. And this misfolding, or this incorrect crossing, presumably occurs during the folding of the ribozyme. And this model is consistent with the biochemical and functional data, but until now, it had not been tested directly because the structure of the misfolded state had remained elusive. So again, we went to Cryo-EM to answer this question. And again, at PNCC, we were able to collect this very nice data of this ribozyme. And this is done under conditions where we knew that we expected to have a mixture of molecules in the native and in the misfolded states. So after building a 3D reconstruction of that data, we saw that the particles were consistent with this very compact folded RNA that resembled the native state of the ribozyme, but there was this density at the core which was not well defined, and that was consistent with a mixture of states with differences localized to the core. Again, we turned to 3D variability analysis, and that variability analysis again was consistent with the core having multiple conformations. And we were able to separate those states, again, using the variability analysis. And essentially, we saw that in our data, our data could be explained by two topology classes. Let's call them topology classes here. And I'm showing here in this close-ups where the differences are between the two separate topologies. One is in this direction of this density here that is showed by this red arrow, right? Where you can see that the densities are basically they are moving in opposite directions. And then this where I'm putting this star here, or this asterisk here, you can see that this helix is rotated. They're rotated relative to each other. So this yellow density here, this #1 volume, was perfectly consistent with the native state, which had been solved by Brijudas and Wachu's lab, right? So that suggested that this other volume represents the misfolded ribozymes, and we were able to then refine those two structures, and then use tools to build a structure into the core of this misfolded state. And for that, we use this very useful tool. It's called AutoDrafter, which I highly recommend to anybody who's working with RNA to look into. This was developed by Riju Das's lab and also in collaboration with Bachu, and allows you to build RNA structures into low to moderate resolution maps, and that you can access that program through this ROSI online server. So this is a Rosetta online server, and there are many other tools there for helping you with RNA structures. Right. So we use these tools to build the RNA structure in the core of the misfolded ribozyme. And when we compare this to the core of the native ribozyme, consistent with this topology model that I show you that was proposed earlier, there is this two strands that are crossed differently between them. This red and blue strands, you can see that in this case, it's the J87. This red one is on top of this blue one, and then it goes under the blue one here in this misfolded state. And what that causes is it results in this rotation of this helix, this cyan helix on the top, which is this helix P7, which contains the guanosine binding site and the active site of the ribozyme. So this topological crossing here ends up rotating this helix. This is a bit again, this is the same thing. I'm showing you here how P7 here is now rotated, and what happens is that now that when P7 is rotated, that basically blocks the active site, such that the substrate that needs to dock into the core of the ribozyme for this splicing reaction to occur is now not possible because of this now steric clash that will occur. So this basically answers the question as to why the misfolded state is not functional, even though they are globally very, very similar. These two states are very similar globally. Now, even though they look very similar, when you just like examine the structure and you think, how much rearrangements, how many rearrangements do I need to make to go from the misfolded to the native state? It happens that it's actually not very simple, and this is it's just a model that I built just by moving things around, just to get a sense of what type of rearrangements you would need to go from the misfolded to the native state. And you see that, let's see if this plays. Oops, there we go. So yeah, so essentially the core has to rotate almost by like 180 degrees to resolve this topological problem. And so that also now answers the question as to why it takes such a long time, right, to go from one state to the other. So in summary, I show you how we were able to use this Cryo-EM to separate particles that were in these different states. And again, this question had been in the field for a very long time, and finally Cryo-EM finally gives you the tools to answer those questions. And our structure shows why our structure basically confirms the topological model that was proposed by Rick Russell and Dan Hirschleich. they were not able to get the specific strands that were, they did propose some strands that were crossed, but they were not able to get that. But that makes sense because they were not, it was very hard to predict this very large rotation of this P7, right? From biochemical and functional data. But it was remarkable that they came up with that model even without having a structure. And that rotation results in this steric clash that, again, that explains why the misfolded state is not functional. And then I show you how it would require very extensive reorganization of the ribozyme to be able to move to the to the native state. And very nicely, after we published our paper, just a couple of weeks later, a few weeks later, this paper came out again from Riju Das and other groups in collaboration, and they essentially made the same observations as ours, and that supports our findings. So that was very nice. So just to close this, I wanted to just go into what the promise of Cryo-EM is for now looking at this dynamic RNA structures that had been elusive for a very long time. We're seeing a explosion really I think it's just going to keep going faster. We've seen all the structures both from our lab and also from Wachu and Riju Das's labs, and there are other groups. And sorry, I forgot to update this slide, but there's many new developments in RNA structure by Cryo-EM. I hope that I also convince you that even at low resolution, we're starting to get some qualitative information about this conformational dynamics undergone by RNA. And we are starting to even start getting information about dynamic processes such as RNA folding. And with that, I would like to go now and to thank my lab, especially my PI, Jeff Keefe, which has been very involved in this project. He's an excellent advisor, and it's a great lab to work at. And I would like to acknowledge Maddie Sherlock and Andrea McFadden for help with the TLS story, and then Quentin, who was part of the tetrahymena ribosome misfolded story, and then the people at Anschutz and at PNCC that helped us with the Cryo-EM data, and then my sources of funding. And if you have any other questions you'd like to discuss further, these are my contact information here. And I want to thank you for your attention.
Thank you, Steve. That was so absolutely beautiful. I'm just like blown away. I have chills. There are a couple more questions. We're coming up on the hour. So if you're okay sticking around, we'll get to the questions there. And if people have to pop off, those will be part of the recording. So find that on our YouTube page. And hopefully we see those of you who'd like to join us again next month, and let us know at the national centers how we can help you use Cryo-EM to solve structures as beautiful as the ones you just saw. So we'll pop back over to the Q&A. Minwoo Sung asks, it's very interesting to see the 3D variability analysis results. Do you have any thoughts on how you could strengthen them using other experiments like assays, MD simulations, or NMR for validation?
Oh yeah, I mean, yeah, I'm really interested. I think that definitely what's the marriage of this variability analysis with Cryo-EM with other tools, quantitative tools, biophysical tools that really probe those dynamics and give you more insight. I think that these are hypothesis generators that you should go and now you use other type of technique. And I mean, I think, it depends on the type of question that you are answering. Like for the TLS story, I'm really interested in those dynamics and of the anticodon arm. And we think that those are related to other functions that the TLS also is involved in. And yeah, I am currently working on some single molecule FRET experiments that I would like to do to really understand those from like a more mechanistic and quantitative way. So yes.
Thanks. Riju Das says, great talk, Steve. Do you think
Thanks, Riju.
do you think Cryo-EM can capture earlier intermediates in the folding pathway for the group 1 intron?
Well, that's a very, yeah, I mean, like, of course, like one of the things that made this easier was that you can accumulate this misfolded state because it's so stable, right? So people figured out ways in which you could basically the solution conditions in which you could like accumulate this misfolded state, so that makes it a bit easier. Now, other intermediates that are more transient, that are they're more challenging. They're like time dependent. Doing some kind of fast freezing maybe, and trying to capture those intermediate states, and collecting a lot of data, maybe to where you can still even see those transient. But yeah, that's what I can think of right now. But I would like to hear more what Riju thinks about this. He probably has good ideas.
Right. Andrew Santiago Frangos asks, using this structural understanding of the misfolded state, do you think it's possible to design a version of the ribozyme that's less susceptible to this specific misfolding?
That's a very good question, and I'm sure that probably that's already been answered somehow. Like even without the structure, probably people figure out ways that you could like that's a very insightful. I'll maybe there is some more insight that one can get from just looking at the structure and the differences between the misfolded and the naive state, and then stabilize or destabilize certain things to kind of like move those arrows, right, that go into one of those in different ways. So yeah, I think probably, but I haven't thought about it.
Yeah, I guess the next question is somewhat related. Have you or others done any modeling of the folding of the ribozyme to understand why that inactive structure dominates?
So those arrows that I show with like 90% and 10%, that can also be modulated depending on the way that you are folding the ribozyme, right? So there's a ways in which you can bias that to move towards the native state. It just depends on the conditions in which you are folding the ribozyme. Earlier work, again, before these 3D structures came out, there's a model for why is this happening, and that is presumably because of this alternative secondary structure that forms earlier in the process. So there's an alternative helix that forms, and basically that traps you into this pathway. And then once that helix forms, then presumably you go into that misfolded state, and then paradoxically that helix kind of like breaks and the original native helix forms. So it's kind of hard to explain, but yeah, so there is but by the question why that dominates, it is just a feature of RNA to form these alternative secondary structures that can potentially trap you in some alternative folding pathway.
All right, there's another question about what is special about the tetrahymeno ribozyme? Why choose that species?
Yeah, it's historically, so this ribosome, the tetrahymeno thermophilic ribosome, was the first, I believe, the first catalytic RNA discovered. So this is when Tomczyk and colleagues, they figured out that RNAs can be catalytic, and that opened that whole field of, and it was a little bit of a kind of, I don't want to say accident, but they figured out that this RNA, they were studying something else, right? And then they figured out RNA without proteins, they can catalyze reactions, and that led to a Nobel Prize. But since then, people started just doing all this biochemistry, biophysics, and all this work on that particular RNA. But there are other variants that also have been used, but I think it's mostly a historical reason. And like now that all these things have been developed on top of it, it became a model system, so it's easier to work with too.
All right, and then Krishna Shivakumar asks, what are the challenges you'll expect when RNA folding involves triplexes and quadruplexes? Does AutoDrafter allow you to build models for RNAs with triplex and quadruplex structures?
I wish Riju could be part
actually,
of the panel.
No, I think I think we can let him talk as well. Here, let me. We'll put him on the spot a bit. I hope he doesn't mind. He should be able to talk. Hi, thank you for joining us.
Hi, Steve. That was a great talk. Thank you.
Thanks, Riju.
I guess for Krishna's question, AutoDrafter is not very good at building models for RNAs with triplex and quadruplex structures right now. So I'm hoping that improvements in the future. Perhaps a lot of it will be spurred by the work of Steve and Jeff Kieft and other RNA structural biologists. I hope we see that improvement in the next 2 or 3 years.
Thanks, Riju.
And if you guys wanted to hop back into the conversation you were having earlier, I'll give you a chance to do that now, because that was our last question in the Q&A.
Oh yeah, Riju asked me if ways to trap or like observe previous intermediates, right? So I don't know, like doing like this, like the similar work like was done with SACs, where you have like a time dependent thing where you maybe that could be a way. I don't know what you think.
I think it's a huge data analysis challenge, and I that's kind of why I was asking. See if folks in the audience knew of. You would require like CryoSpark, to the nth power to be able to like get movies from that kind of time resolved data. But boy, that would be amazing. Yeah.
All right, so I think that wraps up all of the questions left in the Q&A.
Yeah, I have a question that the panelists can't put questions in the Q&A, but I have one quick question about the earlier part of the talk, and then one also about this part. In the TLS and tyrosine acylating enzyme, in the crystal structure, was the same bending of the anticodon the anticodon loop. Was that seen in the crystal structure, and was it kind of perhaps dismissed because of the crystal packing arguments? Did you see the same big bending of that as you saw in the Cryo-EM structure?
Sorry, the bending. What could you remind me what bending we're talking about?
Yeah. There was a crystal structure of the tRNA of the TLS with the acylating enzyme, and that had that same loop. Of course, it was the structures there. That same loop bent by that much to be directed towards the acylating enzyme, much differently from what tRNA does.
So at the resolution that we can, the conclusions that we can make from the resolution that we have, it appears that the acceptor arm, right, is interacting in a very similar way as the tRNA, which we expect because the acceptor arm, even though the secondary structure is different, it does resemble a lot the tRNA. The anticodon arm that it seems to be interacting in a very different way. The loop is approaching the enzyme in a different way. So we don't know how the enzyme is accommodating those interactions. We know that the enzyme, that tyrosyl synthetase enzyme, does not use the anticodon arm as a very strong identity element, suggesting that there's some flexibility there. But yeah, I think solving the structure to higher resolution will probably answer those questions. But as far as I know, there's not much information about that from the crystal structure.
Okay, and then a quick comment about the misfolding in the tetrahymena. It sounds like perhaps RNA didn't succeed as a catalytic thing because there was so much possibility of misfolding. I think what RNA needs is a chaperone for RNA folding, like proteins have for protein folding.
Yeah. So and there's a whole field about that, and I am not probably not the best, but definitely there are RNA chaperones, and I think perhaps they're still a little bit understudied. I know Rick Russell had worked with RNA chaperones and using the tetrahymene ribozyme as a model system as well. So there are RNA chaperones. I don't know how good they are or how they compare to protein chaperones. But yeah.
And actually, there was a quick last question from Krishna. She said who asked about the modeling. Since they're working on RNA triplexes, any suggestions for how to go forward with the modeling? Since we have a couple experts, if you have any tips or ideas to pass along.
Yeah, so I don't have much experience with triplexes. Yeah, I don't know if there are any specific computational tools that one can use for that. Yeah, sorry about that.
All right, thank you. All right, so we've wrapped up with the. Yeah, go ahead.
I'll say maybe if that person can send me an email or something, because I may think of something, or I may find something, and I'll be happy to discuss. So yeah, you can just contact me, and I'll be happy to discuss, even if I cannot help.
Thank you so much, and yeah, thank you so much for the talk, Steve. Yeah, and so you'll let me go ahead and stop the recording.